nerve cell transmissions are electrochemical. So as nerves are sending signals all over our bodies, that's an electrical chemical signal. So how is it electrical and how is it chemical? So we'll take a look at that in this segment, where nerve cells are going to send an electrical impulse, and as they travel to the end of the nerve cell, it's going to release a chemical. That chemical is going to make sure that that electrical impulse carries on to the next nerve cell. And then when that nerve cell terminates at, let's say, a muscle or a gland, then that nerve cell is going to release a chemical to stimulate that muscle to contract, or perhaps that gland to secrete some hormone or something else. So let's take a look at how this electrical impulse is generated. So to start, we need to understand a little bit about just how nerves are normally. So a normal nerve, just doing nothing, has what's called a resting potential. Now this is a slight electrical difference across the membrane. So on the outside of the nerve cell, there tends to be an overall positive charge. On the inside of the nerve cell, there tends to be an overall negative charge. This is a result of the overall charge of ions outside of the cell, the extracellular fluid, as opposed to inside the cytoplasm of the nerve cell. Ions that tend to have a negative charge, large polyatomic ions that tend to have a negative charge. So inside the cell, negative, that's the main thing to remember. Outside of the cell, typically a positive charge. We call that the resting potential, just a slight difference in charge. It's the same thing you'd see in a battery. Two sides to the battery, one side has a slight positive charge, the other side has a slight negative charge. So it's just resting, ready for action. Slight positive charge on the outside, negative charge on the inside. Now this nerve is going to do nothing. There's nothing that's going to happen or change about this nerve until it reaches what's called threshold. The threshold stimulus is going to be enough to excite that nerve and generate that electrical impulse. So you might remember all the way back in junior high how you're sitting behind someone in class and you're trying to put this sticky note on their back, or maybe they're trying to stick it onto your back, and you're trying to do that without them feeling it. Now, if you are successful, that means that you were able to accomplish that task and never reach their nerve threshold, the threshold for their feeling. If you were unsuccessful and they found you were trying to stick something on them, they turned around and they got mad at you or yelled at you, which you probably deserve. But that means that you've crossed the threshold and they've actually felt that. So if you think about anything you feel, like heat, pain, pressure, touch, all those things, a lot of things might operate just below the threshold. You don't feel it. Once it goes above the threshold, the impulse is sent. You get the signal, something's lightly touching your hair, crawling around on your arm, whatever it is. But it just means that the neurons have been stimulated. When the neuron is stimulated, that means these sodium ions with their positive charge are now moving into the cell. So the cell becomes permeable. The sodium ions begin to move in. So all those positive charges now begin to move in to the nerve cell. So they had been outside, now they're moving inside because the nerve cell has been stimulated. It's reached the threshold. It's been excited. So the gates will open, the protein channels, these open, sodium rushes into the cell, causing the inside of the cell now to be overwhelmingly positive. We call this an action potential. It's kind of like a domino effect. The positive ions now rush in, the sodium ions rushing in, they're positively charged, making the inside of the cell now positive, and that's going to cause an action potential, kind of this domino effect. Now that the nerve cell has been stimulated or excited, and these positive ions are rushing into the cell, they're only doing that in this little segment of the nerve. But it's not going to just stop in that nerve segment. That excitability is going to continue down the length of the axon, the long part of the nerve cell. And as it does in each segment, all these sodium ions on the outside rushing in, making the inside of that nerve cell positive. So it's just going to move along this axon, causing each section to become positive on the inside. Now, almost as soon as this happens, there's going to be some ions inside the cell that are going to rush out. So we have sodium rushing in, making it overwhelmingly positive on the inside for just a split second. At the same time, almost simultaneously, we have this potassium ions, which are also positive, moving outside of the cell, taking all those positive charges with it, restoring the negative charge on the inside and the positive charge on the outside. So an action potential is the sudden reversal and restoration of the charges. So we've got sodium moving in. Almost as quickly as sodium can move in, potassium is being pumped out. And as it is, we've got this section of the nerve cell is positive now on the inside, negative on the outside. This electrical change has caused an impulse to start traveling along the axon, this action potential. So sodium rushes in with its positive charges, making the inside positive. Potassium rushes out with its positive charges now, making the outside positive again. So we've got this sudden reversal of charge and then the restoration of that charge, called repolarization, as the potassium moves out. Now we might ask, how? How is it that sodium is rushing in and potassium is rushing out? How does this happen? You might remember the diffusion lessons in chapter 3 and how the sodium-potassium pump would work. Active transport against the concentration gradient. So here we have this large concentration of sodium on the outside. It rushes in. We have this high concentration of potassium on the inside of the cell. It rushes out. Now, this sodium-potassium pump begins to work. It works by pumping the sodium out of the cell, and at the same time, the potassium is going to be pumped into the cell. So see here, the sodium pumped out of the cell, the potassium pumped into the cell, against the concentration gradient, making it so that the sodium gets stacked up here on the outside in a high concentration. The potassium gets stacked inside the cell in a high concentration, so the nerve cell will be back to normal almost as quickly as possible. So we have this sodium rushes in, almost immediately repolarization, potassium rushes out. Then the sodium-potassium pump starts to pump. Pumps that sodium out, pumps that potassium back in. Now we have the resting potential again. The nerve is back to normal. All this is happening faster than a snap. It's going so quick so that your nerve can get yet another signal if it needs one. So all this happens very quickly, sending that electrical impulse through the body. So let's take a look. Here's your nerve cell resting potential, sodium on the outside, potassium on the inside, maintaining that slight electrical difference. Not a whole lot happening here, not a whole lot happening here, not a whole lot. Now here this insect, let's say, lands on your arm. You don't feel it, you don't feel it. It starts to crawl around, moving your arm hair. It stimulates the nerve. Now you feel it. And when you do, you're going to get the whole thing. Sodium rushes in, potassium rushes out. Here's sodium rushing in. Here's potassium rushing out. Here's the sodium-potassium pumping back to normal now. And you're ready for another impulse. This is going to be an all or none. Either the whole thing fires or nothing happens at all. So as soon as you cross this threshold, there's going to be an action potential. The action potential is going to be the same amount each time. And it's going to happen. It's all or none. All or none. Notice how quickly this is happening in milliseconds. So within 4 milliseconds, within two milliseconds of crossing threshold, you're back to normal, ready for another impulse.
So how do you explain the intensity of a feeling Well, it's not the strength of the action potential because they're all the same, but it's the number of action potentials in a given amount of time. The more action potentials in a small amount of time, the more intense the feeling. So if you're being poked really hard or pricked with a needle, that's a lot of action potentials, very small amount of time, the pain is very intense. If the action potentials are spread out over that same amount of time, then the feeling's more light or gentle.
Dixon's doctor completed the neurological exam. He found the median nerve was abnormal, but everything else was normal. The median nerve is located right here in your wrist. What ion channels would be impacted by this compression? If that nerve is being squeezed, compressed, what ion channels might be affected? Well, in the nerve, you have the potassium channels, the sodium potassium pump. All those things might be affected if that nerve is being squeezed. Those channels might not work very well. Now, is Dixon likely to have an electrolyte imbalance? Well, we would think that he would have an imbalance if he was having these nerve symptoms all over his body. But here it just seems to be in his wrist. So it's probably not an electrolyte imbalance, sodium-potassium imbalance. It's probably something to do with that wrist compression. A synapse is just a junction between two nerves. So now we have the electrical impulse traveling along the length of the nerve. It's going to now stimulate the chemical part of that reaction. So here we have neurotransmitters. Those are chemicals. They're stored in the end of the axon. And as that electrical impulse comes to this part of the cell, it's going to open up and allow some of these neurotransmitters to leave the cell, exocytosis, leave the cell now into this little gap right here, the synapse, cross that small synapse and stimulate the next nerve. So you can see these channels right here ready to collect that neurotransmitter, and then that'll cause that impulse to travel along that nerve cell. This happens to be a muscle cell, it would cause that muscle to contract, or if it happens to be a gland, it would cause that gland to secrete something. But this synapse, this little space right here, is just in between these two nerve cells, where this neurotransmitter, this chemical now, is crossing that small subcellular space. So there's all kinds of neurotransmitters, but there's these chemicals that are just crossing that space. Now once they cross that space, the nerve is going to reabsorb some of those neurotransmitters. Others will get broken down by enzymes. Let's take a look at how that works. So here we have the action potential, the gates open, allowing the neurotransmitters now to secrete the neurotransmitter. It's going to open the gates on these cells, stimulating those cells now for their action potential, and that cell will be stimulated. The neurotransmitters reabsorbed, recollected. Some of them will be dissolved by enzymes. Now Dixon's condition could get worse if he continues to work on his laptop because that median nerve will keep being compressed. So the doctor prescribes some ergonomic corrections. How would his compression damage alter his synaptic transmission? So if his nerve is being squished, imagine all this just being squished or pinched in, it's going to really inhibit some of that function. And how might the reduced neurotransmitter release cause muscle weakness? So if we're not getting enough neurotransmitter to cross this synapse and stimulate the muscles in his hands to type, they're not going to be strong enough to perform those functions. Now sometimes on a nerve there are excitatory synapses, sometimes there are inhibitory synapses. What that means is sometimes the neurotransmitter that's released is saying, yes, send this signal, we want to continue the signal along the nerve. Other neurotransmitters are saying, no, we're inhibiting it, we're going to stop this from happening. All right, so sometimes the signal is yes, so we're getting information from this nerve telling this nerve cell, yes, send the impulse. Other nerve endings here are releasing a neurotransmitter saying, no, inhibit the impulse. Slow it down, stop it. All right, so some signals, yes, go. Some signals, no, don't. How do you know what to do then if you're getting mixed signals? Some impulse to that nerve saying yes, some impulse to that nerve saying no. How do you know what to do? Well, you take a vote. Your nerves are democratic, apparently. So it's called summation, where you just add up how many signals from the yes nerve, how many from the no nerve to determine whether the action potential is generated. So let's say that you tally up the votes, 72 excitatory nerve endings, they're saying yes. In this case, we'll say 64 send the impulse that says no. Do you do it? In this case, yes. The action potential is generated, that impulse is sent, that message is going to get to its destination. So let's kind of take a look at how that works. Because there are some other factors. So here we have some yes votes, here we have some no votes. A lot of times it depends on the proximity of those votes, whether or not the nerve ending is sent. So sometimes it has to do with the number, sometimes it has to do with the proximity, like where they are in location to where that's going to be sent. So here you are studying. You're working hard, you're studying hard, and you're trying to figure out, I've got this big test, I want to study for this test, I want to do well on it, but a nice snack, do I do it? Do I not? So we take a vote. In this case, yes, the excitatory votes overrule, and you go to the kitchen, get yourself some cake and milk. Here are some different neurotransmitters that are fairly common in the human body. Acetylcholine is a very common neurotransmitter. It's going to cross between all the nerve and muscle junctions. So anytime a nerve terminates at a muscle, there's going to be some acetylcholine being transmitted at that junction. So its main job right here, causing voluntary muscles to contract. Myasthenia gravis is a problem with acetylcholine. In this case, your body's immune system is attacking the receptors that recognize acetylcholine. So what's the result? The result is that the acetylcholine then, that signal doesn't get to the muscle. So you tell your arm to squeeze, that signal is coming down your arm, signaling your muscles to do that motion, and when it does, it releases some acetylcholine, causing those muscles to contract. But the problem is, without these receptors, the next time you want to contract your arm, it's going to be a weaker contraction. There's not going to be as much of a signal. The next time, even less of a signal, and it'll get weaker and feebler very quickly. That's the problem with myasthenia gravis. Acetylcholine in your brain has a lot to do with stimulating memory. So Alzheimer's, as it's destroying a lot of your brain cells, depletes that acetylcholine levels, which might contribute to loss of memory in this case. So we have this decrease of acetylcholine in your brain in Alzheimer's individuals. Dopamine is a neurotransmitter that's associated with pleasure feelings. So you might think of like, ooh, smoking dope, man. That dopamine is the chemical in those drugs that give you that kind of good feeling. When your body naturally releases dopamine, it's a good feeling kind of chemical. Parkinson's, though, is a disease where the dopamine producing cells in your brain are being deteriorated. So you're not producing the dopamine that you would normally do. In that area of your brain, though, that's where a lot of your muscle control center is. So a person with Parkinson's disease has very decreased dopamine levels because those dopamine-producing cells in their brain being deteriorated, they're not producing it, which sends a lot of mixed signals to their muscles. So their muscles might start shaking or relaxing and contracting or stiffening up depending on what's happening at the time. Norepinephrine is a neurotransmitter that is always antagonistic to acetylcholine and usually associated with good feelings. So they're kind of like a brother and sister, where if you have one, you would recognize how you always do the opposite thing. They say, I like chocolate. You say, I don't like chocolate, even if you really feel that way, just to be antagonistic, just to get under their skin, just to do the opposite thing that they do. So whatever the situation is, these two will be opposite. So if acetylcholine is saying yes, norepinephrine will say no. Norepinephrine is saying no, acetylcholine will say yes. So they're always opposites. But generally speaking, norepinephrine is associated with some good feelings. Feelings of excitement and arousal and just general good feelings.
Serotonin is a neurotransmitter that is thought to have a lot to do with a generalized feeling of well-being. So again, it's kind of good feelings. And a decrease in serotonin is connected often with depression and signs of depression. So loss of serotonin leads to wakefulness, not enough serotonin, often feelings of depression.
Endorphins are painkillers. So these are naturally produced in our brain to override some pain sensors, and again, to kind of help us feel better. So if you're ever involved with like runners or you know any runners, a lot of times what they'll do is they'll run and run and run and run and run and run and run. It gets to be very painful, but as they push themselves past that pain, their brain releases these endorphins to help override those pain centers, and they get what's called a runner's high. So they get these good feelings as a result of running and putting their body through this kind of painful experience. Similar things would happen with like acupuncture or other things that help release painkillers, these good feelings to kind of override our pain centers. So let's review with a little bit of matching and see how we do here. Wakefulness, which these has to do with wakefulness. In this case, serotonin. The opposite to acetylcholine, they always do the opposite. In this case, norepinephrine. Pleasure feelings, in this case, dopamine. Potassium rushing out of the cell. All right, so when potassium rushes out of the cell, that's going to be repolarization. When sodium rushes into the cell, that's when we get our action potential. When sodium's normally on the outside, potassium's on the inside, that's our resting potential. When we're combining the excitatory and inhibitory effects, that's going to be summation. We're just going to add those up, see which is the larger number. Acetylcholine is a neurotransmitter that controls voluntary muscle. And that's an overview of Chapter 7, nerve cells and how nerve cells work.